Our next speaker is Dr. Jean Hebert. He's one of our extramural researchers. He's a professor at the Albert Einstein College of Medicine in New York. And his presentation today is Progressive Brain Tissue Replacement and How We Beat Aging. You can read his presentation summary and bio on the events website. And now let's welcome him to our stage.
Hi, everyone. Thanks so much for having me. I'd like to acknowledge the SENSE Research Foundation in several ways. One, for their support of our projects aimed at reversing brain aging, but also for the excellent students that they've sent our way. Recently we've had Sanjana, who's presenting a poster tomorrow. And so really excellent students. So thanks for having that program and letting us participate in it. So, there are a couple of things that we work on in the lab. One is a microglial replacement project, and I think I've talked about that in the past. So I'm going to focus a little bit more on a brain tissue replacement approach, and why I think that that's really important for defeating not just brain aging, but overall aging. And I'll give you my reasons why, and then I'll be happy to discuss and take questions for, if anybody else has any ideas or especially critiques. So definitely open to critiques, and then being able to make this a better project. So the reason that we're thinking at the tissue level of replacing brain tissue is that really the damage that accumulates with age occurs largely outside of cells. So a lot of the focus on cellular processes with the epigenome, with mitochondria, with genetic reprogramming, et cetera, might make the cells perform better and the organs overall do better. But the bulk of aging that's occurring outside of cells is complex, it's stochastic, and it's not enzymatically driven, and it cannot be enzymatically fixed. So there are no genes encoded in our genomes that can address this damage. So examples are shown here on this slide, just for proteins. There's also carbohydrates and DNA that accumulate complex damage. And so there's no way for at least the extracellular protein damage that the cell can repair that. And without that environment, the cell behaves as an old cell, even if it's young. So in heterochronic transplants, where you take young cells, put them in an old brain, they behave like old cells, and vice versa. If you take old cells out of the brain and leave them in culture, neurons, for example, they can outlive the age of the organism. Or if you put them in a young environment, then they'll do better as well. So I think it's really important to think of, if we want to beat aging, to do that at a tissue level, tissue or greater level. So there are other people working on replacing, other parts of the body, either as a whole or progressively with different parts as you need them. But in any case, those are not what my lab works on. But in conjunction with those, I think there's a lot of promise here for actually beating aging. So is brain tissue replacement possible without a discontinuity of self or function, right? Because that's important. The whole point is to preserve who we are and continue enjoying life. So I think the answer is yes. And I'll give you the reasons for that. We're focused initially on the neocortex, which is the outer part of the brain. It's the biggest part. It's arguably the most important. It encodes our long-term memories, cognition, highest cognitive functions, consciousness. So, if we can't fix that, then, what's the purpose of working on anything else, really? So the two principles that support that progressive neocortical tissue replacement is possible is, one, plasticity. So memories and functions can seamlessly move from one part of the neocortex to another. And I've given this example before, but it's only one of many examples. where language, something that's very dear to us as a function, can move from one area to another if that initial area is slowly destroyed over time from a pinpoint out. In the case of a benign brain tumor, for example. So these individuals, eventually have the tumor removed. They have a big hole in their brain, but they never lost the ability to speak. And no one ever noticed any difference. They didn't notice a difference. So they're perfectly fine. And then when they go look for where speech is encoded, it's somewhere else in the neocortex. Not always the same place, but it has moved. So this is a level of plasticity that tells us that we can replace tissue in the neocortex without losing continuity of self. The other sort of established principle that supports progressive brain tissue replacement as a means of beating aging is that young neocortical precursors, when you transplant them into the adult environment, don't seem to care that they're in an adult environment and follow their innate developmental program and do remarkably well, even in the adult brain, to wire correctly, even to distant targets in the brain. So this is something that's been reproduced by many labs. We've done some work with this as well, both with mouse into mouse graphs, but also human into mouse graphs. So here's a human example where we have the human graft in green here, and it projects its axons in the correct orientation down to this major tract here, the corpus callosum, which you can see is bright green, and that's because of all the projections coming from this new brain tissue going along these tracks. And if we look further at more distant targets in the brain, we see that those neurons are innervating those areas. And I'll give you a little bit more detail on that in a bit. But this is just to say that these cells, these precursors of the brain, have this established potential to generate the connections potentially that we want in the brain. And so, I mentioned other groups have done this. These are examples of high-profile papers. In this case, where they all use human precursor cells, neocortical precursor cells that they transplanted into adult rodents. And all showing all these pretty amazing properties. And many of these are from different labs, so highly reproducible. So, the tumor example I gave with the plasticity, if done right, means that we can remove old tissue. And the example I gave you and these other examples from other labs show that you can add potentially new tissue. Certainly the cells seem to do very well in the adult if they're immature precursor cells. So we should be able to add new tissue that can encode information that's useful to the host and allow function to transfer, allow self-identity to transfer without us even noticing. But not there yet. So despite those very nice examples where connectivity of transplanted cells is remarkably good, it's not good enough. There are things missing. One thing that's missing is these graphs typically don't contain the full complement of cell types that are normally found in the neocortex. And all the cell types are there for a reason. They're all doing something important for function. And without them, the tissue can't function normally. So that's one thing, is they're missing cell types. Another thing is the organization of the cells is very abnormal. So if you look at, one of these acclaimed successes of transplanting organoids into the brain, you can see that the tissue is very different than what you have in a normal tissue, where you have this very nice layered structure. That is not recapitulated. Maybe in micro locations it is, but that's not good enough to get normal function, because the normal lamination is important to get normal wiring within the graft. So that's two things that are missing. So our goal is to address these missing aspects of grafts so far to make grafts that will develop into a normal structure and will have the full complement of cells. And to do that, we don't want to reinvent the wheel. We're basing our approach on how our neocortex normally developed from a relatively simple fetal tissue at around gestational week 6, where there's a limited number of precursor cell types in a very well-defined organization that we want to recapitulate in a proto-tissue that we will then use to graft. And then with the anticipation that this tissue will develop the correct layers. And as already shown for disorganized tissue, will project normally outside of the graft as well. So this is an example of a couple of key aspects that have been missing from all grafts so far. In green here, there's a small number, but a very important pioneer neurons called Cajal retius neurons. They're very important for setting up the architecture and the wiring of the neocortex, the layers that come afterwards. Also very important are a stable radioglial fiber attachment to an overlying layer here that's not shown, but I'll come back to that. So these fibers guide new neurons. So these radioglial fibers are part of the radioglial stem cells that are not only stem cells, but by virtue of these fibers act as scaffold for the new neurons to migrate and form these layers in the neocortex. So here's sort of where we're at with this project. This is again the prototype tissue the proto-tissue that we want to make. It has 3 layers, this pia layer, this marginal zone, ventricular zone, doesn't matter too much for your purposes what they're called, but they have relatively few numbers of precursor cells in them. And all these cells can be derived and have been derived by us or our collaborators from human iPS cells, except for one we're still working on. And then we can also purify all these cells, again, except for the one that we haven't managed to derive from iPS cells yet. Sorry about the noise, if you can hear it. And so it's not just the cells that are important here, it's also the extracellular environment that are in to make sure that these cells kick off the right developmental program from the start. So we're doing proteomics to identify the extracellular components that surround these cells in each of the different layers. So this is an example of the transcriptomic, single cell transcriptomic analysis that we do to compare our iPS-derived neocortical precursor cells. This is for the principal neuronal lineage, and we compare that to real human fetal neocortical tissue and find that we get a very good match. And we do this for all the cell types, again, just as sort of a quality control. And so we've also established a platform, which we've published now, showing that we can get, the major features that we want in testing these proto-tissues in the mouse, in the adult mouse. So we can create these very reproducible lesions that we can fill with our precursor cells and biogel. And then we find that all the cell types that we care about survive in this graft. And the neurons integrate. They send projections along normal axonal pathways like the corpus callosum, the major fiber tract that connects the 2 hemispheres, down to subcortical targets like the striatum. More importantly, they also seem to mature normally. Initially, there's very little activity, but over time, these human neurons start to show action potentials as they normally would. And moreover, if we put the graft in the visual cortex, over time, the neurons will respond to light. So this, hopefully you can see this little orange arrowhead here is the onset of the light stimulus for these mice. And you can see that the neurons in the graph initially after 4 weeks are weakly responsive, but then become more robustly responsive over time. We also, very importantly, have vascularization of the graph. So we include vascular endothelial cells in our graphs. Here they're labeled with GFP, so we can see them in green. They rapidly form vessels. Those vessels fuse with invading host vessels. And you can see an example of this anastomosis here. And if we put a circulating dye in the animal that goes throughout the whole system, we can see that that dye goes through these green vessels as well, not shown here, but I may have shown that previously. So we have this platform that's pretty good. We can also layer cells in this platform. Again, these are not the layers that we want yet. They're just a proof of concept that if we use a biogel that's commercially available, very popular one that supports neuronal survival, we can layer green GFP labeled cells and then overlay, I think in this case it's TD tomato labeled cells, and this border is maintained, but blood vessels can still cross, as do neuronal projections, which is the properties that we would want. So we have this platform now that we can test with tissues that are more and more resembling the normal fetal tissue. But for that, we have a little ways to go. We can isolate pretty much all the cells, as I mentioned, -1. We've made quite a bit of progress on identifying key components in the extracellular matrix to make the biogels that these cells are going to initially be embedded in. And then, of course, there's going to be, a fairly extensive phase of testing whether the tissue develops into these mature layers, neuronal layers, and whether it can encode function. So we have outlined experiments that will prove that the neuronal function of the graphs, their electrophysiological activity, is encoding useful information to the host. And then if we can do that, then, success. But we might need to do a few iterations before we can do that. So that would fall into the bigger project here, a bigger phase that comes afterwards, where we're not only adding new tissue, but we have to remove the old tissue. So we're going to want to mimic what I showed you as an example with the benign glioma that grows slowly from a pinpoint out and destroys different functional areas. Of course, we don't want to use gliomas because when benign, there's a risk that they will become glioblastomas, and that's definitely not worth the risk. So there are other ways that we can do this that we've designed on paper, but these have to be developed in reality, where we use an optogenetic approach to start silencing areas from a pinpoint out. And as long as the individual is using those functions, they will get re-encoded elsewhere, and they won't notice a difference. And then once that tissue is electrophysiologically dead, we can remove it. So that's shown here with the silencing of this tissue. So this is our prototype tissue that we add in. The brain shrinks with age, so we can add some tissue without removing anything initially. But then we have to remove the old tissue, and that's what's shown here with the method I just described. And then once that's removed, we can add the new prototype tissue. It will also grow in, and we repeat this. And in a few iterations, we have a completely young neocortex again. And this would be combined if we wanted to really beat brain aging, which we really do want to do, but don't have the resources to be working on these other parts of the brain yet. We would want to do this in combination with other parts of the brain, particularly the ones outlined here. So we need more people working on this, basically. And we already have, a lot of collaborators and a lot of people in the lab who have worked on this. I don't know if Sanjana is in the audience, but apologies, I didn't update this acknowledgement slide, but Sanjana has been a big help here as well. Yeah, and also to try to accelerate things, here's a shameless plug for a company I've started where we're basically focusing on trying to develop this tissue initially to treat patients, but it would be applicable to addressing aging, and combined with replacement of the rest of the body. I think that we can, in the foreseeable future, actually beat aging, not just live healthier lives as we nevertheless age, but actually beat aging and then decide, when we've had enough or keep going as long as we want. Okay, so let's see. Guess I can stop sharing, and hopefully I left some time for questions.
Okay, Dr. Hebert, fantastic, exciting, and provocative as ever. Thank you very much. I am so delighted we started funding your work. Unsurprisingly, we have a whole bunch of questions. The first one comes from Dr. Sharma, who's an immunologist, and therefore he asks an immunological question. He says, the old brain tends to have senescent cells, such as in the glia, and also infiltration of T cells. How do you think their presence might affect the engraftment of the new tissue?
Yeah, totally a valid question. We have to go with what we know so far, which is even with naked cells transplanted in humans to treat Parkinson's, for example. So, back in the 80s in Sweden, they started using dissociated fetal cells from midbrain to treat Parkinson's. And those, in post-mortem analyses, showed no signs of no pathogenic signs for at least a decade and a half. And then in one case, they had a patient that lived where they collected the brain 24 years after the transplant. And those were starting to show pathology. And we know these brains are inflamed, but those neurons were still functioning quite normally until for a long time, decades, and those were naked cells. So our expectation is that cells that are in a new tissue, so they're not just naked cells, will be first more insulated from any negative effects of the old brain environment, but also that this is over time we're going to replace all the parts of the brain so that it will be completely young. So I think our tissue will certainly do well enough for long enough for that process to occur. That's the expectation. Thanks.
I think you have a more optimistic read on those Parkinson's transplant studies than I do, but we can have that argument another day. I think he was actually asking a question that was more proximate, which is like not will the cells survive for decades once they're implanted, but when you initially are trying to implant these, what do you think is the effect of the presence of, in aging, there is already infiltration of T cells, there is already senescent cells, etc. If you're trying to do this in an aged brain, how do you suppose that's going to affect the initial engraftment in the first place?
Yeah, again, we have not done enough in mice, but maybe more has been done in humans already with these, examples of Parkinson's transplants. These are not young people that are getting these cells, and they seem to be surviving and doing quite well. There's a very recent example from Blue Rock Therapeutics, where they have these wonderful dopaminergic precursors. They're in phase 1-2 trials, and they're getting positive effects, not just for safety, but efficacy after, I guess now it's like 10 months. So it's still early stage, but I think there is reason to be optimistic, but I'm very happy to discuss potential challenges and caveats.
Yeah. Another thing I would add on just the immune side of things is, of course, that if one was working with iPS cells, that eliminates the allogeneic concern. So that actually was a question that I think addresses one of Yafei's question right there, which is you could, if you start with iPS cells, which is actually what you're doing in your animal experiments for the most part, you avoid the allogeneicity problem. He also asks, when considering the aging brain, it's apparent that the entire organ may be affected. How can transplantation of a portion of brain tissue to enhance cognitive function? And I think this question sort of answers itself, but so let me ask it a bit more broadly than he's asked it. It's imperative that the entire organ may be affected. How are you going to handle areas outside of the neocortex where you're focusing?
Yeah, that's a very good question too. It, undoubtedly, the whole brain is affected, and undoubtedly, every part of the brain is important for function. And so we need to address all parts of the brain. Again, we're going to be, I think, guided, although again, we haven't started for the other parts of the brain, but I believe we're going to be guided by normal development. Um, and so, for example, the visual thalamus would be replaced at the same time unilaterally as primary visual cortex, for example, so that they wire together the same way they would during development. Um, but yes, like, you could replace the whole neocortex, but then you'll get a stroke into some subcortical
Mhm.
structure and that will affect function, or just things will stop working.
Yeah.
And so you don't get, proper information flow from the outside world to your conscious cortex. So yes, yeah, those all have to be addressed. So this is a monster project. Yeah,
Yes,
I
sir.
hope I make it sound like it was simple. But the takeaway too is that the result is a monster result too. I mean, I think we
Yeah.
can beat it, right? So that's, you got to weigh the pros and the cons here.
Yeah, yeah. Well, and of course you had to start somewhere. Literally, I mean, you had to start somewhere. And so, like the neocortex is a very high value target to say the least.
Right.
So yeah, but absolutely a perfectly intelligent question. Like as you say, if you get a massive damage in a subcortical structure, it's not going to matter if your neocortex is functioning.
Right.
Um, I'm going to keep asking questions until someone throws me off. So, um, since Jason Zhao asks, since the brain will continuously atrophy over time and each surgery will replace only a small percentage of the brain, what is your best estimate of how often brain tissue replacement surgeries would need to be done in order to keep the brain healthy indefinitely?
So I think we could replace the entire brain with like a half dozen to 10 surgeries, which sounds like a lot, but again, that's not something that's not doable. And surgical techniques are getting better and better. I haven't been keeping up with surgical robots, but they're making a lot of progress, a lot faster than I thought. And that reduces complications of surgery. So there is risks with surgeries, of course, but I think those will be diminishing and a little more controllable. So yeah, 6 to 10 surgeries over the course of 20 years, and then you basically have a young brain again. I don't want to say like a teenager brain again because that'll scare a lot of people. But you have a young brain that will last for decades, and then you don't have to do anything for a long time.
Yeah. I will also add from the SENS perspective that once you've done that, you can slow down the further progression of damage by doing all the other damage repair strategies that SENS has. So once you have a nicely pristine young brain in there, it's going to be a lot easier to maintain it by doing things like removing amyloid and alpha synuclein aggregates
Yeah, yeah,
and direct molecular level repair of the extracellular matrix. But if you've gone and replaced it wholesale the way you're doing, that's a very, very systematic approach to say the very least.
yeah.
If I'm still allowed to ask you questions, Laura Lynn asks, are there any concerns about the invasiveness of multiple brain surgeries?
Yeah. So I mean, I kind of addressed that a little bit there, and I think Maria's appearance here is telling us we have
You
to
can
wrap
answer
up.
that question, and then yeah, we have to wrap.
But it is. There are complications from brain surgeries. Not many deaths, because you can actually do away with a big part of your brain without dying. But there are complications, and those we'd want to avoid those. And so surgical techniques that are minimizing that are being progressively improved upon. And at some point, I think the risk will be low enough in a short time. I mean, these are AI-driven smart robots, and I think progress there that'll be ready by the time we're ready to go, I think.
Super. You've got another good question and a good comment and another question I'd like to ask you sometime, but I will get off and people can perhaps approach you privately if you're hanging around. And thank you, as always, for a great presentation, Dr. Hebert.